We have the neural crest cells being able to migrate, okay, into the mesenchyme of the developing dermis, and they will differentiate into melanoblasts, which will later differentiate to melanocytes, okay, and then we'll have some granules embedded in them. Okay, and they do this at the junction of the dermis and then the epidermis. So we call that the dermo-epidermal junction. Dermo-epidermal junction. So we want to use this diagram to explain what I just said. So that is the epidermis. The dermis are the dotted black spots. And then the blue ones are the migrating neural crest cells, which are forming melanoblasts, and then which will later become melanocytes. You should appreciate the difference between the melanoblast and melanocyte by now. The difference with the blasts and then the cytes. You know that the blast is an undifferentiated cell, so it can still continue to grow or divide. Then when it becomes differentiated and mature, it becomes the cyte. So you can see that these blue dotted areas are migrating and moving.
Hello, sir.
Yes.
Please, can you say the melanoblasts are the precursors for melanocytes?
Yes.
Okay.
Yes. So if you want to compare it to an adult and a child, you can say melanoblasts are children who are still growing compared to an adult who is no longer growing in height. You have matured. Okay. So these blue ones are laying themselves down at the junction of the epidermis and then the dermis. And you can see they are releasing some red particles here. So those will become the granules we are talking about. What is the function of the melanocyte? Anybody? So it
Sir, they are
yes.
for the production of melanin, the skin
Good,
pigment.
good. The skin pigment. So these are the granules I was referring to, the pigment granules. Okay. And these melanocytes can form dendritic spines, as you can see. So from the basal layer, they extend. And remember, this basal layer also marks the junction between the epidermis and then the inner dermis. So these spines or finger-like projections they are exhibiting is known as the dendritic spine. And this also gives us the 5 or 4 layers of the epidermis: stratum corneum, stratum granulosum, stratum spinosum, and then stratum basale or germinativum. You also have the stratum lucidium, okay, and it occurs in a particular type of skin. There are 2 types of skin. You have the thick skin and then thin skin. So the stratum lucidium is found in one of them. I want you to look out for which one exactly you find stratum lucidium. So in the epidermis, you have these cells. We've talked about melanocytes. You also have what we call Langerhans cells, and then you have Merkel's cells. So the melanocytes, as we have said, are derived from neural crest cells. Okay, and they appear by the 8th week. They will get to the epidermis. Between the 4th to 6th months, they become dendritic and they synthesize and transfer melanosomes, which will contain the pigment granules. The Langerhans cells are derived from bone marrow, and we know bone will develop from mesoderm. So in fetal life, the yolk sac or the liver performs a similar function, okay, of the Langerhans cells. And then you have between 6 and 7 weeks, they appear and then they mature around the 12th and 14th week. The Merkel cells, some say is derived from neural crest cells, others don't really can pinpoint. So evidence points towards origin from primitive ectodermal cells within embryonic epidermis. They appear in the plantar skin around the 12th week, and then by the 16th week, they appear in the palmar skin. Okay, so it means that they appear earlier in the lower limb than in the upper limb. So when you have this diagram, periderm, which we know came from the ectodermal cells, which became the basal cells. Then you have keratinocytes also forming, which will later help form the keratin, topmost layer of the skin that helps to protect it. So usually we say the skin is stratified, squamous, keratinized because of these keratinocytes. So I've put here, compare the activities of melanocytes and melanin in white and black races. Why do white and black people look different in terms of color? And compare and contrast thick skin and thin skin. So you remember I asked a question relating to another layer of the epidermis known as the stratum lucidium. So look for the thick skin and thin skin. You will know which one contains that. So again, the keratinocytes are there, and then you can see that the ectodermal cells are now invaginating. Okay, so the basal or stratum germinativum is budding downwards. Basal cell bud primordial hair follicle. So the hair is about to form. Okay, so this is how the hair formed. That we go into details on that. So having talked about the epidermis a bit, let's look at the dermis. If you look at this diagram, it's a vertical section through the embryo. This is around the third week where you have ectoderm, mesoderm, and the endoderm. That is the yolk sac. So in the mesoderm, this notochord is marking the midline. So flanking the notochord on the left and right are these mesodermal, points. You have paraxial mesoderm, intermediate mesoderm, and then the lateral plate mesoderm, which splits into two. The splanchnopleuric or the visceral mesoderm, and then the somatopleuric or the somatic mesoderm. Okay, so most of these visceral mesoderm will form a lot of the visceral organs, and then this will form most of the muscles, okay, of the body. So remember that this somatic mesoderm will end up enveloping the body during lateral folding. Okay, when the body folds laterally, you end up having the visceral one inside, and then the somatic one will encircle the body, while the ectoderm also covers the body entirely. So this is what I'm talking about. So that's the ectoderm. So the ectoderm is covering, okay, the entire body. Within it you have the mesoderm. So the mesoderm has these somites, okay, and the somites also can develop along two lines. You have this dermatomyotome going epasia, that is upwards, or hipasia, downwards. So some of the muscles will grow downwards, others will grow upwards. Okay, and then some will also be stationary. And these two are the intermediate mesoderm, which I pointed to here. Okay, right. So dermatomyotome is there, and mostly the somites are going to come from the paraxial mesoderm. This one, the one which is very close to the notochord, paraxial mesoderm. Then you have intermediate mesoderm, and then you also have the lateral plate mesoderm. So when you look at this diagram too, neural tube is here, just as you have neural tube there, okay, and the neural crest cells in green. So here, that's the neural tube. Neural crest will be flanking the sides of the neural tube. That is the surface ectoderm. Okay, the same surface ectoderm that thickened to form the neural tube, and then the neural tube will now cut itself off from the surface ectoderm. So the surface ectoderm will reassume its original, form. by covering the entire body. And like I said, during lateral folding, the hitherto ectoderm, which is flat at the top, will end up enveloping the entire body. Then within the mesoderm too, you have this dermatome, you have a myotome, and the epiphyseal muscles moving up, epiphyseal going down. Sclerotome will help form some of the skeletal structures, okay, and then cartilages. That's the notochord. Then you also have angiogenesis going on in the dermis, because we know that the epidermis is usually avascular. Okay, so you need the cells beneath it to help with diffusion of substances towards them in terms of oxygen and food. So the blood vessel in the dermis begins as simple endothelium line structures known as angioblasts. So again, you see a blast has formed, so it's young. Okay, so some capillaries acquire muscular coats through differentiation of their myoblasts. Myoblasts will be the young cells of what? The muscles. Okay, they are not differentiated yet, so they can also divide and then will form myocytes later on when they are mature, which will become muscle cells. And those obviously will be smooth muscles. So the capillaries that have these muscles will become arterioles and arteries, because we know that capillaries need to allow for exchange of substances. You don't expect them to have any thick muscle coats. They should have just an endothelium and then the basement membrane. All right, so just as you saw the ectoderm, and for that matter, the epidermis invaginating to form the hair, glands of the skin also go along that line. So we want to look at sebaceous glands. Most sebaceous glands develop as buds from the size of developing hair follicles into the surrounding connective tissue and branch to form primordial of several alveoli and ducts. Okay, so the alveoli are actually the spherical portions of the gland. Okay, usually that is where you have the secretory unit. Then the ducts are the passageways for the content that the gland has produced. So if you have sweat, you have a sweat duct. And sebaceous gland also has its duct that it passes through, the oil. So if we take this to be, remember you saw something like this earlier on the hair follicle formation. You can see this blue area. So that's the sebaceous gland also starting its formation. It's always linked to the hair follicle. So it secretes its substance to the hair follicle and then it comes up. So you also have on the right here another type of gland known as the eccrine gland. Usually these are the sweat glands. Okay. They do not secrete into the hair follicle, they secrete directly onto the surface of the skin. The central cells of the alveoli break down, that's the sebaceous gland, and an oily secretion known as sebum will mix with the disquamated peridermal cells to form venous caseosa. Those who began with me, you remember I mentioned this venous caseosa and said we'll get to it. So that's where we've got into. So the venous caseosa is sebum mixed with disquamated peridermal cells. And we know that the peridermal cells are the first layer of cells that form on top of the ectoderm or the basal cells. So disquamated means that they are squamous cells, but they have been roughed off, as in peeled off. Okay, so that's the disquamated peridermal cells. So when they mix with the oil, they form sebum. And usually for a baby that is born, you see that thing on her or him, like a glassy substance covering the whole body. So that is it. So these are the sweat glands. The eccrine ones also develop by invagination. So they develop as epidermal downgrowths into the underlying mesenchyme. As the buds elongate, their ends will coil to form the primordium of the secretory portion of the gland. The epithelial attachment of the developing gland to the epidermis forms the sweat duct. So that will be the duct, and this will be where the secretory portion will be. This is not showing the coiled part, but there's a diagram coming very soon that you see the coiled nature. The peripheral cells of the secretory part of the gland differentiate into myepithelial cells. So the myepithelial cells are specialized smooth muscle cells, okay, which are usually found in glands. They contract and relax to allow the substances to be pushed out of the secretory areas. So this is a summary of the sebaceous, what do you call it, the sweat gland. That's the eccrine sweat gland, bud of sweat gland. Then it goes down and then it's starting to coil. Then you have the coiling there, and you can see that it opens straight onto the surface. Okay, so that will become the duct, sweat duct. And then you have the secretory portion. That's the myoepithelial cell, the specialized smooth muscle cells. Okay, and that's also for the hair. We looked at that to also invaginating or budding downwards, and then you end up having all that. And then also associated with it is the sebaceous gland we looked at. So we said that sebaceous gland will secrete into the duct of the hair follicle and then onto the surface. There can be some abnormalities of hair development. When you have so many hair on the body, how do we call that? There are some people who have a lot of hair, which is abnormal. How do we call that? Anybody who knows that? If you don't know it, then you go and read abnormalities of the hair. We also have apocrine sweat glands. We have seen eccrine sweat glands and apocrine. These ones are usually related to the armpit region. Okay, and so you have them in the axilla and then the pubic area, and then also the perineal region. They develop from downgrowth of the stratum germinativum. We have talked about stratum germinativum as being one of the layers of the epidermis, which is always vibrant, divides and forms new cells on top of it, okay, to replace those at the top. As a result, the ducts of these glands open not onto the skin surface as do eccrine sweat gland, but into the upper part of the hair follicle, okay, because they are coming from the stratum germinativum, which also gives rise to the hair follicles. We are now talking about breast, which is also part of the skin. So they are modified sweat glands. So it's funny. Breast that people are crazy about is nothing than just a sweat gland which has been modified. Okay, and these mammary glands begin as buds. And even before they form the bud, they form crests, and then they form buds. So this is how the crest looks like, and it's all the way from the axilla to the groin area, which means that this whole area is a potential breast formation. But eventually, it is only the one close to the chest that remains. The rest will vanish. But there are certain abnormalities that occur that the rest will not vanish. They will still be there. And so sometimes you find, the breast persisting, forming ridges, and then the breast will develop along that line. So we are saying that you have primary bud, which will later also bud to give secondary bud that will now form or develop ducts known as lactiferous ducts. These ducts or the secondary buds will canalize to form the ducts. If we say canalization, we mean creating a lumen. So canalization of the secondary buds to form the duct is induced by placental hormones. Okay, the fibrous connective tissue and fat of the mammary gland will develop from surrounding mesenchyme. So this is what we are talking about. So primary mammary bud, primordium of the mammary gland. So that is the bud. Then you have secondary buds also coming from the primary ones. Okay, so these are extra extensions. Then you also have the canalization of some of these secondary buds to form the ducts. So this is what is going on. So that's the lactiferous duct, which you open here. Site of depressed nipple, areola of the breast, the ducting area of the breast. Okay. So you can see that this is a depression, but as the child grows, it will now be elevated. Okay, instead of depressing. So during the late fetal period, the epidermis at the site of the origin of the mammary gland becomes depressed, forming the pit. The nipples are poorly formed at that point. At birth, the nipples usually rise from the mammary pit, okay, because of proliferation of surrounding connective tissue. So this is what we are talking about. So you can see that it's now pointing from A all the way to F. It's now protruding. So the breasts of newborns contain lactiferous duct, but there are no alveoli, okay, because obviously they are not producing milk. In females, the breasts enlarge rapidly during puberty, mainly because of development of the glands and accumulation of stroma and fat. The lactiferous duct of male breasts remain rudimentary through life because we don't use it. Milk will not pass through, so it remains rudimentary. So the abnormalities of the breast, like I told you, from the axilla, that's the armpit region, all the way down, you can see some of the points showing that nipples are potentially there. Okay, this at least you can see this one and that one because of the crest, the mammary crest that runs all the way down there, which is a potential site for development of nipples. So you can have polytelia, okay, and then polymastia. So you should know the difference between what I've mentioned, the two, polytelia and then polymastia. Now, still with the skin, we can have nails. So fingernails and toenails begin to develop at the tips of the digits as nail fields at approximately the 10th week. And they do this at 4 weeks interval. So it means that if one is 6 weeks, the other will be 2 weeks. Okay. So the fingernails and toenails at 4 weeks interval respectively. So nail fields will then migrate onto the dorsal surface. That's from the ventral surface. They will go to the dorsal portion, carrying their innervation from the ventral surface with them. Nail fields are surrounded laterally and proximally by folds of epidermis. So you can see this lateral fold, and this will also be some of the proximal folds. So that's the proximal fold talking about, and that is the lateral fold. This is the nail field, and that's the nail plate. So the nail plate, the arrow is showing the development, is going to grow on top of the nail plate. I mean the nail field. The plate is developing on top of the nail field. And have this eponychium above and then the hyponychium below here. So cells from the proximal nail fold grow over the nail field. So this is the proximal nail fold, and then the nail plate is developing. Okay. So you also have abnormalities or anomalies of the fingernails. You read on that as well. So this will bring us to the end of the lecture. Those